Well, I'm from academia. I was there for 20 years at USC and at Children's Hospital Los Angeles, but things were going slow in there. By the time I finally get my machine, it would be completely outdated and useless. So I went into biotech after that and was involved in working on trying to get next generation sequencing, which eventually went to whole genome sequencing into clinical practice. And now I'm the medical director of doing that, and it's in New Jersey, but it's actually on Zoom across the country and the world to take patients with autism and with other neurodevelopmental problems as well, and to use whole genome sequencing to try to figure out what's wrong with them and to help them. And I'm going to show you some of the data, and then I also am a founder of a company that takes those findings and translates them into new products, and that's NeuroNeeds. So I was asked to talk about the genetics of epilepsy, and what I'm going to be talking about is the genetics of epilepsy, but also how it fits in with autism. I have 4 slides, so I'm going to not go over the data very quickly, but these are the largest studies that I have seen in the world in which they have looked at epilepsy, and they've taken whole exome sequencing, which is all of the protein coding sequences. Remember, exome is only about 1 to 2% of the genome. So these are looking at about 1% or so of the DNA to sequence all of the genes. I'm not aware of any whole genome sequencing studies. I'm going to show you some preliminary data on that. This is the largest study by far, almost 10,000 with epilepsy. They showed that GABA pathways are very important, and I'll get to that. In the parentheses is my text. You can see some of the other pathways that are involved in epilepsy. Here's a smaller study, 125, that they were able to really take a very good look at the DNA in this. And you can see that there's GABA receptors again, dendrite development, gene expression, all different things here, including ubiquitin ligase. We'll get to all of these pathways. But the thing is that there's a pathogenic or likely pathogenic variance in 40%. Here's another one, 103, epilepsy 72, autism 14, and then they found only about 24% for epilepsy, 28% for ID, for intellectual disability. The highest diagnostic was for epilepsy subtypes and early onset seizures, which makes sense. And then they found that there was a higher one for exome than there was just for a panel, but it was not significant. And then this one right here from the Netherlands, 100 patients, they had an IQ of 85 or less, as well as epilepsy, and then there's a 40% rate. So the literature, it varies depending on which the patients are. There's many other studies out there that have looked at like groups of a particular syndrome or whatever, but they're generally finding between 25 and 40%. Here is just to show you that, we're talking about variants. Nobody wants to be a mutant, so we're not supposed to use the word mutation anymore. A mutation is a genetic variant which is associated with disease, but that's basically, we're now saying disease-associated variants, which is a little bit harder to say. Variants in the across the horizontal axis can be small, a single base pair. Most variants in the genome, whether pathogenic or benign, are a single base pair, but they can be as large as millions of base pairs, chromosomal abnormalities, and anywhere in between. And they can be as common as 50%. If you have a variant which is in 49% of the population, the 51% is considered the normal sequence. Of course, that's if people actually identify the one that's more common and what's your population you're using. But anyway, the variant is the minority one, and then the most rare variant of all would be a de novo variant that's only seen in that patient. It has never been seen in anyone else before, including not in the parent. So when we're talking about doing DNA sequencing to look out for clinical, you have risk factors can be extremely rare, and they can be extremely common and anywhere in between. But then causal variants that actually cause the disease, or at least are the lion's share of the disease, are presumably going to be rare. So when we go through these variants, and one of the things is going to be gene frequency, think about a risk factor can be at any frequency, and a causal one is going to be, by definition, it's going to be rare, or else it would be causing disease in a whole lot of people. This is the basic science talk. You probably know what an Illumina sequencer looks like. It costs $1 million. It can do a whole genome sequence in 4 hours. The big difference between these sequencers have been around for a while, and whole genome sequencing has been done by a few people for the last 4 years or so in clinical studies. The big difference is the computer algorithms. When you're looking at 3 billion base pairs, in particular, the average person has about 40,000 genetic variants in and around the genes. You need really good software that can take a look at that. And so every single year, there's been larger amounts of the genome that have been looked at. 10 years ago, we were doing panels. Panels were like 1,000th of the genome. Then we were doing exome, that's 1 to 2% of the so now, in my clinical practice, I'm doing 99% plus of the genome, basically all of it except for some very difficult areas which are highly repetitive that really don't show up very well. So we've increased just in 10 years from 1,000th of the genome to the whole genome. And then, of course, in the 10 years prior to that, there wasn't any DNA sequencing at all. So the sequencing has improved, but it's also the computer algorithms and, of course, the databases. So this is a moving target. If you just go back 2 years, a study that's based on data that was collected 2 years before that, that's extremely ancient data. So I'm going to show you data just from 2022. It was started to collect it in January, and I stopped collecting it in August. So it's already dated to some degree. And it's only 20 patients, but this is the main difference is that there was a major computer change in the way that the data was looked at that occurred about October. It took me a few months to get good at it, so I said, okay, January was where I'm going to start. So I'm taking a look at the 20 most recent patients that I've seen. It's a telemedicine practice, so it's nationwide. You can see the ethnicity is mostly Caucasian. They all have neurodevelopmental disease. They all have a clinical diagnosis of autism, and it's sort of a loose, part of that. And they may or may not have other problems, but they all have a clinical diagnosis of autism and neurodevelopmental disease. They all had TRIO, that's including the parents' whole genome sequencing that was done at Variantyx, which I don't have a conflict with, but I think it's a great lab. And it was, they were analyzed this year. And so the first thing is that of the 20 patients, 15 of them had a monogenic diagnosis that was causal. The numbers have been going up. Every single year I find more and more, but now I'm at 75%. I don't think it's going to go up any higher because I think the other 25 are polygenic. These are small numbers, but as you can see, of the 15 that were monogenic, 7 of the 15, slightly less than half, were listed on the laboratory report. Most of those were listed on the laboratory report as, take a look at this may or may not be important. But occasionally they were on the laboratory report as, we're just going to show you a couple of other things, and this is sort of not as a diagnostic at all. 8 of the 15 were nowhere on the laboratory report. I did the raw data analysis myself. I had 5 years of doing that at Cortigen, and I was briefly at Lineagen, and I've been at 2 other companies as well. And so I have unlike most physicians, I have the skills to be able to do that, and I will go through some of this data. Here's some other things. I put down at the bottom, because this is on seizures, that if the 4 of the 5 that had seizures that we were able to make that had a monogenic diagnosis, and then the 2 out of 2 that had subclinical spikes. So overall, for seizures or spikes, it was 86%. Not statistically significant because of small numbers. One other thing is that if the mental retardation was on the mild to non-existent, and the patient had tics, I found 0 of those. Those are most likely PANS, PANDAS, or whatever you want to call those. But if the patient had significant mental retardation and no tics, I found essentially all of those, one exception. So I think that, in the autism population, there are a lot of them that are actually more immunological. That doesn't mean we didn't find genetic findings. That just means they're polygenic. Here are the de novos. There's 15 of them. And the color coding is on board. I made the colors so that the red, green, yellow, and blue are treatable pathways. Now, some of them are genetic mechanisms, the ones that are in gray. Those are not treatable pathways. And then there's an immunological pathway in purple. But all of the other ones are treatable. So at the top, you see there's the de novos, and then there's the inherited. 2 of them were autosomal recessive, and 2 of them were X-linked. And then these are the 15 with causal diagnoses. And again, I want to emphasize that most of the pathways are treatable. There are 3 of them on there that you see that they're underlined. The 2 of the greens and the 1 purple, they're very large, like thousands or millions of base pairs. The others are single base pair or a few base pairs. And you can see that there's the same pathways that a lot of people are talking about: oxidative stress, including mitochondria, protein turnover, cation channels, and amino acids. The usual pathways. Because I'm going to be talking at a seizure one, the reds have seizures and the greens have spikes. But anyway, so that they're kind of all over the place. So slightly over half of them, the laboratory didn't find it. Why? Are the laboratories completely useless on this? I was a medical director of Cortigen for 5 years, and I was the one that was missing them. So I'm not knocking the labs by any means. The difference is that it is my patient, and I am the genomicist at the same time. So I know the patient far better than the lab did. I've had a relationship with them. I've spent hours talking to them. I've seen the kid, and at least telemedicine. But most importantly, I can go back and ask questions. What do the ears look like? Is there a family history of bleeding disorder? What did the MRI show? What was the potassium level in the blood? I can go back and ask these questions and say, okay, we can eliminate these, or oh, this one I thought was nothing, but this is more important. I can order more tests. The big difference is to be able to do that. So, okay, so there's 7 of them that were missed. And 2 of them, the one at the top was an atypical. The phenotype was extremely atypical for what's written in the literature. So that makes sense. 2 of them were extremely rare. One of them, there were 2 cases in the world. The other one, there were 4 cases in the world. So that didn't get to the laboratory saying, well, this is a known disorder. But 5 of them are new disorders. 5 of them have never been found before. This is the first case ever. So how do I know that? How can I prove it's a new disorder? Well, they were all de novo. Did I find other de novos? Certainly there's de novos out there. There are other patients with de novos. These 5 de novo, and some of them are probably causal, but I'm not calling them that because I have no idea. These 5 are causal because these 5 are all in genes that have been implemented in autism before. It's just nobody has found a case to report. 2 of them are known because there are SNPs in that gene that are associated with autism. 2 of them are known to be associated with autism because there are functional studies that have shown that that gene expression is abnormal in autism. And 1 of them is in a pathway in which a gene very close to it has been associated with autism. And it's the gene that's immediately adjacent to. So 4 of them have direct connection with autism, and 1 of them has a very close indirect connection. So I was able to make a clinical diagnosis, plus the other parts of the phenotype fit. It made sense. Okay, so that's the 15 that had monogenic diagnosis. What about the other 5? Well, the first one there is a really weird patient. It has episodes of shock. Yeah, the kid has autism too. The kid's normal, intelligent, has episodes of shock, and the sister died of a similar problem. This is really a very different phenotype. Has a de novo mutation that is in a gene that's of uncertain significance. So I can't count it as a hit. Probably is, but I'm counting it as a miss. Have no idea. There's one that has MEFE that's in the inflammatory cascade. That's a PANDAS patient, a PANS patient. So it makes sense it's in an autoinflammatory gene. By the way, I find autoinflammatory genetic mutations in a lot of patients that have PANS and PANDAS. And the other one here, there's a mutation in the GABA receptor, and there's a mutation in the immunoglobulin chain. This is one with immunodeficiency and with autism, probably polygenic. Another one has a calcium channel abnormality, and clinically that fits the rest of the phenotype. And then there's the last one there that is no clue at all. I found absolutely nothing. So overall, 15 cases out of 20, 15 cases had a causal diagnosis, and then you can see the breakdown there. And then of the other 5 cases, 3 of them I'm pretty sure I know what the problem is, but it's polygenic. But I can't prove it, of course, with polygenic. But overall, I think the most important point here is that of the 20 cases, 16 had clinical changes to the management based upon the DNA. So, I mean, I'd already put them on mitochondrial cocktails. I had already made sure they were on folinic acid. These patients, we had already looked at their diet and their sleep and all that sort of stuff. I'm not talking about these things. These things are critically important in clinical management. But they had changes that were made based upon the genetic findings in 80% of them. So this is not just, this is a gene XYZ or something. You can't do anything about it. Sometimes that is the case. But out of 20 patients, 16 had changes based upon it. And then another thing is, it's like insurance companies and people are saying, well, you're going to find all this other stuff that's off target and we don't want to know it. 2 of the 20 had off-target findings that had nothing to do with their neurodevelopmental disease. In this particular one, they were both von Willebrand disease, which is a bleeding disorder. It's an extremely common bleeding disorder. If you get into a car accident or have a major surgery, you can bleed to death, and then there's an antidote for that. One of them was later found not to have von Willebrand's disease. It's probably a benign polymorphism. The other one was found to have von Willebrand's disease on additional testing. So it's really only 2 findings, and then one of them was followed up and actually has something off target. I know that I go fast, but a lot of it has to do with ion channels. You guys all, this is a basic science audience, you know what ion channels are. There's many different ion channels. That was a very common issue. Ion channels are common problems in epilepsy for sure, and they're also common in autism. How much time do I have? Am I already over? How much?
You have 2 minutes.
Okay, 2 minutes. I just want to go over this one girl really fast because 13-year-old, she had already been given a diagnosis of autism since she was 7 years of age. Had auditory and visual processing disorders as well. At 13 years, she had a respiratory virus. She lost all of her academic skills within 1 day. She was a normal student in a normal class. And then she went to that. She also became regressed behavior and acted as an infant. It was almost like a fugue state. And she had weird seizure-like events. Everyone said it was epileptic, but the EEG was normal. They were always preceded by a headache. They were with muscle weakness, more on the left. She had ataxia, tachycardia, pallor, urinary incontinence, clammy hands, and was nauseous. The EEG, like I said, even during one of these events was normal, and of course they said that she was faking it. She's faking the schoolwork too. She doesn't want to go to school, so she's acting like a newborn infant. Anyway, she also has migraine and a few other things, and frequent infections. And she was on high-dose steroids and IVIG. We did clinically, that is hemiplegic migraine episodic ataxia. And there are 3 genes which are known to cause that. Calcium channel A1A, calcium channel A1S, and the ATP pumps. The calcium channels, of course, allow calcium in. The ATP pumps sodium and potassium respectively on each side of the membrane, and then that gradient is used to pump calcium by another pump. So these 3 genes are involved in basically intracellular calcium formation. So which one did she have? She had variants in all 3 of them. It's probably why she had a really severe case of disease. Some were inherited from mom, one of them was inherited from dad. So, and each of the variants was extremely rare in humans and very highly conserved. And put her on a mitochondrial cocktail and put her on potassium chloride and acidozolamide. That was the she dramatically improved. In February 2020, her academics was at a kindergarten level, so she had gotten better on non-specific therapy. She went from infantile to kindergarten level, first grade in math. A year later, the same test was done. She was 8th grade in reading and 7th grade in math. She was an 8th grade student, so she had basically caught up completely. Her regressed behavior had better, and she became age appropriate, which of course she was acting now like a 15-year-old girl and driving the parents crazy. But her ataxia resolved, the POTS resolved. She still has some chronic pain and fatigue, and that's what we're working on now. But I mean, there are some cases in which the diagnosis is not just this is what it is in the treatment, but the treatment can make a dramatic change. And then I'll stop here. Synapses, you all know synapses are important in autism, and then ubiquitization, which is protein breakdown, is also very important. Angelman syndrome is one of those disorders. And so here's my information. So